MetaGenomics can provide additional insight into the function of complex microbial communities and their role in host health. Therefore, MetaGenomics has the potential to advance knowledge and solve practical challenges in a wide spectrum of fields. Whole genome shotgun sequencing, WGS, has been increasingly recognized as the most comprehensive and robust approach for metagenomics. It offers the advantages of identification of species-level taxonomy and the estimation of metabolic pathway activities from human and environmental samples. Several large-scale metagenomics
and associated bioinformatics approaches. Today, ion torrent sequencing systems coupled with the Ion 16S metagenomics kit and the Ion Reporter software is the only single-source end-to-end workflow that enables the rapid identification of microbes present in mixed research populations, both at the genus and species level. According to the journal Wound Repair and Regeneration, 6.5 million individuals in the United States are affected by chronic wounds, and over $25 billion are spent annually on treatment. Clinical researchers are now investigating ways to analyze microorganisms which may potentially help improve lives in the future. The Ion 16S metagenomics solution allows clinical researchers like Dr. George Watts to explore with greater depth into their work.
I'm George Watts, and I am an associate professor at the University of Arizona, and I run the Genomics Shared Service, which is located in the Arizona Cancer Center. Diabetic foot ulcers arise due to the complications of diabetes. The problem the doctors are facing is if they don't know what species of bacteria are present in the ulcers, it's very hard to treat them. So we're beginning to do the clinical research that would allow us to make an improved diagnosis of the species that are present in the wound and the species that are causing problems in the wound. One of the best ways to study and understand the microbiome of diabetic foot ulcers is by looking at the diversity at a DNA level. If you culture a diabetic foot ulcer, you'll see maybe up to half a dozen species of bacteria present. But if you do sequencing-based approaches, you can see pretty easily a dozen to even a few dozen species. So with this much more refined view, we're very interested in doing the clinical research to determine if this diversity has any effect on patient outcome. With a better diagnosis, we would then be able to have better antibiotic stewardship and also better treatment, more effective treatment by giving
bacteria from complex samples. The Ion 16S metagenomics kit enables robust data by minimizing culture bias and by including 7 of the 9 variable regions in their analysis to help deliver fast bacterial identification from naturally occurring mixed uncultured samples. Dr. Charles Lee, director of the DNA Analysis Laboratory at EMSL, uses the Ion 16S metagenomics kit, the Ion PGM, and Ion Reporter software.
And how it is performed. First, let's begin with what the 16th ribosomal RNA is. Lewis and his colleagues first described bacterial RNA genes as molecular clocks, due to their uncommon features such as universality, activity and cellular functions, and extremely conserved structure and nucleotide sequence. Bacterial taxonomic classification, because it is more easily and rapidly sequenced, and contains enough phylogenetically information. The 16S ribosomal RNA gene consists of 8 highly conserved regions and 9 variable regions across the bacterial domain. The degree of conservation varies widely between hypervariable regions, with more conserved regions correlating to gene sequence similarities. It is usually used as the gold standard for taxonomic identification at the species level. A range of 0.5% to 1% sequence divergence is often used to delineate the species' taxonomic rank. Sequencing the 16S ribosomal RNA gene is currently the most common approach used in microbial classification as a result of its phylogenetic properties and the large amount of 16S ribosomal RNA gene sequences available for comparison analysis. RNA gene is universally distributed. Second, abundance of 16S RNA gene sequences exceed those of other bacterial genes. Third, it can be used to measure phylogenetic relationships across different taxa. Fourth, horizontal gene transfer isn't a big problem. Fifth, costs to perform 16S operation, sequencing, and data analysis. After DNA isolation, the DNA is selectively PCR amplified, using primers targeting the 16S ribosomal RNA gene. Common next-generation sequencing platforms cover 100 to 600 base pairs per single read, with varying degrees of accuracy, but the full-length 16S ribosomal RNA gene consists of approximately 1,500 base pairs. Therefore, primers are chosen to cover only a portion of the 16S ribosomal RNA gene. There are several primers pairs codecs for various sequencing systems, are shown in the figure. Because the V1 to V3 region has been identified as the most useful region for distinguishing among species of the ubiquitous and clinically important skin bacterial genus Staphylococcus, this region is generally used for skin microbiome studies. 16S ribosomal RNA-based sequencing is currently conducted using several technologies, including large-scale clonal Sanger sequencing, Illumina-MiSeq, 454 pyrosequencing that targets specific regions or is linked to barcoding strategies, and BioSmart sequencing. When sequencing is performed with Illumina-MiSeq, V3 and V4 region is commonly amplified by using.
Good morning everyone, this is Jyoti Jaglan from Guru Jameshwar University, Hisar, Haryana. I'm working on antibiotic resistance from wastewater and its characterization. I'm trying to do a workshop on metagenomics from last few years, but there is no suitable workshop that is regarding my work. So I trying to check on further, but then I see in December about BDG LifeSciences. I enrolled for the workshop, and then after joining the workshop, I get a excellent experience about hands-on workshop. It is very helpful in my work, especially because I am working on antibiotic resistant genes. So it is helping in my work and further. Thank you.
Hi, my name is Tanu Bhakchandani. I am a PhD student at Jawaharlal Nehru University, School of Biotechnology. I am working on HIV with Dr. Ravi Tandon. So my work includes metagenomics, and this workshop has really helped me to understand the various basics in it, and I'm looking forward to many such workshops from BDG. Thank you.
Hello everyone, my name is Ankita Rai. I'm doing PhD in biotechnology from JNU, New Delhi, and right now I'm in the first year of my PhD, so my work hasn't started yet, but I might work on metagenomics in future. So this workshop has enhanced my understanding on metagenomics, like the 16S rRNA sequencing, and I hope this knowledge will help me out to carry out my PhD work in future. Thank you so much.